Hello friends, so we are here with another video and this is the introduction part, as you can see, and this is all about a Indian fruit, but it is all Indian favorite fruit, but it is all found in all over in the world, and it has many names. And this is called in Cuba it is called Zapote, in Venezuela it is called Nespero, in Caribbean it is also known as Nasberry, in South Florida it is also known as Chico Sapote or Chico. And in India it is known as Chiku or Sobeda. Sobeda, Sobeda. So this is a very sweet fruit, a little bit different in taste, but it's very tasty. This fruit is very tasty. But in my lab it has arrived after getting affected by a fungus. So we will be discussing about this in the latter half of the video, and we will be going to the lab for the analysis of this analysis of this fruit, or the fungus analysis of the fungus. We will see it under laparoscope, and then we will excavate it, and we'll put it in the culture media, and therefore we will start the first investigation, and then the fluorescence study and the mycotoxin extraction and all that. As from the appearance, it has been seen when when I got that specimen, it was white sponge-like. It was there, sponge-like appearance it was having. So I thought it is a sponge fungus, but let me see what fungus it is. It is looking like a sponge fungus, but it is maybe something else, or maybe something it is not. Maybe it is a sponge fungus only. So that we will be seeing when we will go to the lab. So let us go to the lab and see the specimen. So friends, we are back in the laboratory, and as you can see, the two specimens of our specimen, the Chiku, we call it Chiku. Many names, as I have said in the introduction. So here you can see these two beautiful fruits, and how now you can see the condition of these two beautiful fruits. They have become more beautiful after acquiring fungus. So the fungus has showing its own beauty. So here we will be exploring them, and first of all we will see the fungus. We will actually see the fungus under laparoscope. We are going to see the fungus under laparoscope. Closely we will watch the morphology of the fungus on the laparoscope, and then we will go to the hair strips, the culture, pressing them in the culture media, and then we will be going for the other investigations as I have done in my previous videos. So let us look back in the laparoscope. So you can see the fungus under laparoscope, as you can see. So this is more or less looking like white sponge, but it may be something else that we will be getting under microscope when we will see under microscope. This is the protruding structure. This is the posterior side of the chiku. So you can see as a fungus is protruding out. This is sponge fungus. It is protruding out. It's a more or less is a sponge fungus. So it is protruding out. So you can see the protrusion of the fungus here in the laparoscope, and this is the other sides. As you can see, these are the spots and the sponges. So these are the spots and the sponges, and you can see many sponges, many spots, white spots. This is a white flex actually. This is actually a sponge fungus and a flex fungus. This is actually happens when the fruits or the vegetables gets overhydrated, and some petrification signs also you can see under microscope. You can see this started petrifying. They started petrifying, so that you can see. So this is the condition of the chiku. So another one is there, another host is there. So you can see the next host. This is the condition of the host. This is the full petrification. As full petrification, you can see the whole fungus has become, whole chiku has become petrified. It has changed. The normal color has changed. The normal color of this chiku has changed, and it has petrified. And you can see the acquisition of the fungus over here. And see, this is the anterior side. This is the top. This is the top portion of the chiku, and it has changed. So this is the anterior side. Anterior side has got a bulged up. It is bulged up, and the shape has changed also due to the fungus, and it has become watery inside. And we will cut open also, and then we will see the fungus. The fungus is almost on the surface, But don't don't think that the fungus is not entered inside. It has already entered inside. This is because this type of fungus has very big roots, so they have already started excreting their mycotoxins inside. So the fungus is like this. So this is that it has become red. Almost this portion has become red. So this is the fungus. This is the condition of the fungus. Two hosts. So now we are going to cut open and we see this is the posterior side. I can see the more white portions over here. You can see this is the white portion of this chiku. So this is the condition of the fungus laparoscope. This is the condition of the chiku under laparoscope, as you can see. So this is the more or less the host. So this is more or less the host. This is more or less the host. So we are now ready to excavate the fungus. As you can see, the total host is there. So we are going to excavate the fungus, and as you can see, we are just with a scalpel and a forceps we are going to excavate the fungus. This is a very soft, the host is very soft actually, so it won't be difficult to get acquire a fungus. So excavation will not be very difficult. With the help of a scalpel we can excavate the fungus. Just upper skin, we will take out the upper skin and the fungus is out. So a little bit host should take, we should take the host also, and the fungus is out. And the fungus is out. And some other portions we will be taking. Some other portions. The posterior side is much more affected with this white. So let us see how much we can take. This has started acquiring the fungus actually. This has started acquiring the fungus. The fungus has started acquiring. So here is the host. We have to cut the host and just with the help of the tweezer, forceps, with the help of the forceps, the fungus is out. So just putting inside. So this host is over, we can leave it. And this one, the second one. The second one is the main portion. We will take this petrified portion, this big one, this petrified portion. We will take this one and see something is there or not. So this is the petrified portion, and just cutting off the rest part, the maximum part, the cutting of the maximum part. We will take only the small, lesser one. We will take the lesser one, this one only, and it is over. So the rest over, rest of the chiku is to be left off. So here is our excavation is complete. Excavation is complete. So we need to take the culture media, and with the culture media we will be putting it. Now all set, so we are ready. Now we are going to put the culture media. So here is our pipette, and a sterile pipette needs to be there, and by taking a sterile pipette, and we are here using sodium acetate differential agar, because as I have told you in my previous videos, this agar is very beautiful for this type of sponge fungus, everted fungus. This fungus which protrude out this eversion, this is everted fungus. So this type of strong funguses, this sodium acetate differential agar is the best agar. So we are going to put a little bit amount of sodium differential agar, so around 5 ml, 10 ml differential agar. So this is around 5 ml over here, and another 5 ml. and this is another 5 ml. So mostly 10 ml is enough for everything, for a culture. So it is now over. So it is now over. So here we will put back and some of our tissues here. So we will take and some sponge. So our culture is over. Culture part is over. So our host is ready. Our culture is ready. So we will be back after 15 days or 10 days. Let me see how long it takes to protrude or to grow the fungus growth, how long it takes. So after that we will be back. So bye from now. Hello friends, so we are back with our after 48 hours. Remember, we are after 48 hours, and you can see the culture. Well, it has changed the color. The medium has changed the color. This usual color of sodium acid differential agar is green. Now it has become yellow. So you can imagine the fungus is so fast growing. And presumably, which I told you in my previous part of this video, that it is a sponge fungus, but no, it is not a sponge fungus. It is much more dangerous fungus. The actual name I will reveal you later. It is better known as, commonly it is known as Sooty mold. It looks like this white appearance. It is white appearance, but it is not. This is its initial appearance. It is not white. It fortunately it becomes after a few days, when it matures, it becomes black. So it will become black. So now let us see the first stage or the first initial investigation of this medium. As you can see, lots of sporangiums and sporicides are there, or fringes are there. So let us begin with the first investigation. So with the help of a photopipette, we are going to take around 1.5 ml, and so here is our 1.5 ml, and this is almost clear, so we can just take it and let us kill these bubbles. Okay, so this is our 1.5 ml, almost 1.5 ml or 2 ml, like that, and this is enough for our first investigation. So we will be going to the microscope and let us see what happens. So let us go to the microscope. So we are under the microscope, and as you can see the soot, and this is actually a fungus. The name, actual scientific name of this fungus is Capnodium Magnifere. It is commonly known as sooty mold. So this fungus is Capnodium fungus, and it is a very bad type of mold fungus. It causes a digestive problem. It causes digestive problem, and it releases the micropore or microspores or sporangiums, which gets airborne, and it causes lungs diseases, many sorts of lung diseases. As you can see, the pulmonary hemorrhoids. Perhaps you know a disease called pulmonary hemorrhoids, and that is hemorrhagia. That is caused by this type of funguses in infants, mainly in infants and old age adults. So this is a very bad fungus. Initially, you can find this is Capnodium Magnifere, and this is very bad. So this is commonly known as sooty mold. So this is the first investigation, and first investigation is very bad investigation, very bad. So this is a very powerful fungus. It grows very well, and it will make the whole vegetable black. So this is all about. So you can see the presence image of the Capnodium Magnifere or the Sooty mold. So this is actually the structure of the mold, with inside you can see the mycotoxins in different luminescence. It is full of mycotoxins and is a very dangerous fungus, as I have told you. So this is the fluorescent structure of the mold. So we are trying to see some other parts of the slide, and let us see some other molds. These are the similar molds we can see. Some similar molds we will be able to find out. And this side there is nothing, but let us see something. These are the two molds. These are the same structure of the two molds. These are the similar molds of the Capnodium Magnifere, or it is commonly known as Sooty mold. So this is the structure, and so some other portions of the slide let us see. So another mold is coming. So this is another mold. This is the shape of another mold. So the microscope is getting adjusted to see the mold properly. And this is the fluorescence image. You can see the mycotoxins inside burning like a light. So these are the mycotoxins. Generally they absorb the fluorescence and they emit light according to their wavelength. So that is the chemiluminescence of the mycotoxin. So this is mostly the part of the mycotoxin, part of the structure of the Capnodium Magnifere. So it is a dangerous fungus and can disrupt the digestive system of human being, and also it enters in micropores or spores inside the lungs, and it carries the mycotoxins group of trichothiocin group. So you can see the UV spectrum. So two types of mycotoxins are revealed here. One is Verrucarin and another is Calonectin. These are the two trichothiocin group mycotoxins which you can see. One is at 332 nanometer and another is at 374 nanometer. Very bad mycotoxins.